dear friends from abroad and from domestic. Welcome to the Youth Development Series of the 5th World Laureate Forum. My name is Yangang. I come from the Center for Excellence in Brain Science and Intelligent Technology, Chinese Academy of Sciences. I will serve as the moderator of this session. This forum brings together the laureates as well as outstanding young scientists to promote communication and understanding between scientists of different generations, and with the hope to inspire new academic ideas and promote scientific progress. The theme of this session is on life science and ecology. The topic of this session will cover from structural biology to neurobiology to conservation ecology. We'll have two parts. For the first part, we'll have each young scientist to take turn to give a 5-minute presentation about their latest research. And after each presentation, there will be a 5-minute Q&A. The laureates are welcome to give comments and ask questions. After all the presentation, we'll go to the second part. We'll have about a 50-minute long panel discussion. First, it's my great pleasure to introduce our distinguished guests, four laureates. The first guest is Dr. Leif ANDERSSON. He's a professor of functional genomics of Uppsala University, Sweden. He was among the first scientists worldwide to apply molecular genetics to domestic animals. Has been awarded the Wolf Prize in Agriculture of 2014. Welcome, Dr. ANDERSSON. Our second guest is Dr. Stefan. He's director of Max Planck Institute for Biophysical Chemistry. He received 2014 Nobel Prize in Chemistry for the development of super resolution fluorescence microscopy. It's very good to have you, Dr. Hell. Our third guest is Dr. Avram HERSHKO. He's a professor of Israel Institute of Technology. He received 2004 Nobel Prize in Chemistry for the discovery of ubiquitin-mediated protein degradation. Welcome, Dr. HERSHKO. Our fourth guest is Dr. Edvard MOSER. He's a founding director of Calvary Institute for Systems Neuroscience and the co-director of Center for Neurocomputation of Laureate University of Science and Technology. He received the 2014 Nobel Prize in Physiology or Medicine for the discovery of cells that constitute a position system in the brain. Welcome you all. It's a great honor to have this to have you join us for this exciting forum. Now we'll go to the first part of this forum, and we'll have the four young scientists to have a short presentation of their research. Our first speaker is Dr. Demeng Jiang. He is the postdoc from College of Urban and Environmental Sciences of Peking University. Dr. Jiang, are you there? You can start to share your screen and start your presentation.
Thank you all. Thank you, Professor, for the introduction. I'm very honored here to present works that conducted by Dr. Fang Yuanhua, and myself, and all collaborators. The topic I'm going to share today with you is achieving co-benefits of biodiversity conservation and ecosystem services in forest restoration. As we all know, forest loss and degradation have become serious problems, and a lot of environmental problems also. Forest restoration is a promising way to address those problems. because forests provide multiple functions such as biodiversity conservation, water yield, soil erosion control, carbon sequestration, and timber production. A lot of restoration projects have been launched, and many hectares of trees have been planted. Among these planted forests, there are major two types. One is plantation, another is native forest. Plantations consist of one or few tree species and have very simple structure. Native forests, however, consist of many species and have complex structure. Given the differences in structure and composition, maybe the performance of ecosystem service are different. So to answer that, we, Dr. Feng Yuanhua and her collaborator collected a worldwide data set. They found that compared to restored native forest, plantation delivered a significantly less in species abundance and marginally so in soil erosion control, and not significantly in carbon sequestration. However, they found that tree plantation have a clear advantage in tree plantation in timber yield. Oops. Given there's a clear trade-off between tree plantation, timber yield, and the ecosystem services, it's crucial to find a solution for them. In the coming slides, I will use biodiversity as a proxy to explore the solution. In other words, we will ask how to reconcile biodiversity conservation with timber production. To meet timber targets, one way is to manage forests in a biodiversity-friendly way, which referring land sharing strategy. However, conserving biodiversity in plantation may reduce timber yield, therefore resulting more land being used for production and less land being used for conservation. There's a contrast solution. The contrast way is to manage the forest without considering biodiversity, which referring a land sparing strategy. In this strategy, all plantations are dedicated to produce timbers, therefore would have high yield. Therefore, will result in more land, less land be used for production and more land for conservation. However, it's not clear which strategy is better in reality. To answer the question, we conducted a study at Guangxi province, southern China. We selected 4 types of forests that vary in timber yield and biodiversity, and we investigated birds, plants, bees, and estimated timber production. Our preliminary results suggest that species richness of birds decreased dramatically with timber yield, with monoculture have the highest timber yield, but lowest species richness. However, at landscape scale, we found that most species have higher total population when the land managed under sparing strategy. To wrap up those 2 projects, we conclude that to achieve co-benefit of biodiversity and ecosystem services in forest restoration, we should to restore native forest on non-production land and plan land use at regional scale between production and non-production land. That's all of my talk. Thank you for listening.
Well, thank you, Dr. for this wonderful presentation. Now this is open for comments and discussion.
I could comment. So this is, of course, a very important project that you're doing, and because of the loss of forest and loss of biodiversity worldwide. Maybe one comment I could make is that another aspect one could take it into consideration is also loss of genetic diversity, because when you plant monoculture or also what you say this cultured forest, so to speak, that they often have a very limited genetic diversity. So another advantage of what you suggest here is that you maintain genetic diversity among trees for the future. So I mean, I got this Wolf Prize in Agriculture, but I worked quite a lot with biodiversity also. For instance, we are doing studies of the evolution of Darwin's finches, and we had a paper this summer where we showed that adaptation in this species is to a large extent based on genetic variants that have been maintained for very long periods of time. So our conclusion from that study was that it's as important to maintain diversity in common species because they have the possibility to adapt in the future, and that the genetic diversity depends on genetic variation that have been accumulated over maybe a million years or something like that. So
yeah, thanks for the comments. I think it's a very fresh goal for me. Yeah, I totally agree with you that it's also important to conserve those common species. One thing I could add to that is for those common species, I think we have enough habitat, or they are very adaptive to our environment. So at current situation, probably the priority is to conserve the species that sensitive to disturbance. Yeah. That's my prediction.
But what you can say is also that even common species, if they become isolated from each other, then you get small population that is losing genetic variation, and you may lose important genetic diversity that is important for the future, so to speak. So I think we shouldn't forget it depends, of course, how well they spread between different areas. So
yeah,
but fragmentation of the biodiversity is an issue as well. So
yeah, I totally agree with you.
I mean, this sounds great what you're doing, but an important question is, of course, how well government and local administrator take this into account. What is your experience in trying to implement these things in practice?
Yeah, that's a very good question, and I think first of all, the study in this area is quite new, especially in forestry. Actually, the land sharing sparing strategy framework is dominated from agriculture, and my research is pretty new in forestry. If we get a final result, I think it's worthwhile to inform the government how to manage our land. Yeah, that's a definitely important way, and we should definitely do that in the future. Thanks for the suggestion.
I could also make a comment that we have a similar discussion in Sweden, where I'm from. So Sweden has a lot of forest. Sweden is not so densely populated with 10 million people or 10.5, I think, and we have a lot of forest, but a lot of this forest is maintained for timber production and pulp production. So forestry is, in fact, is a very important part of Swedish economy, and there is also always this conflict between producing maximum amount of timber versus maintaining biodiversity. So I think the work you're doing has relevance also for discussions in Sweden, I would say.
Yeah, you have mentioned a very important aspect. we actually also considered the net income of the timber yield. We actually have conducted some preliminary analysis because we haven't done the analysis yet, so I didn't put the result in this talk. Yeah, that is a very interesting and important aspect. We will do that analysis for sure in the future. Yeah, if you can give us some suggestion in the analysis, I think that would be wonderful.
I mean, you are welcome to contact me with questions. I will be happy to discuss with you.
Thank you so much for the option.
Yeah. So I could tell you, I also have an affiliation with a university in Texas, and there I'm worried that I don't think they take sufficient care of the environment. So when I give talks at this university, because they have a big golf course which is a part of the university, and I used to say that they should, because there's so many golf courses in Texas, so you don't need a golf course at the university. So I recommend them to take down the golf course and make it into native forest as an environment, which I think would be wonderful for faculty and students to have a chance to walk in a natural forest close to the university. And it will be these type of places, which will be a place where migrating birds could stay and natural habitat could develop. So I think there is many places where we can do better in maintaining biodiversity.
Yeah, totally agree. Yeah, different forest types support different type of birds, for instance. So yeah. Each type of forest are important. So in this sharing sparing framework, we actually can add more types. So because of the time issue, I didn't add that result in this talk, but yeah, so I agree with you.
Good. Thank you.
Thank you, right. Prof. Andersson.
I see Dr. Moser has raised his hand. If you have a short question, please go ahead.
Yeah, I had a very naive question because I really don't know anything about forests, but you compared the plantation approach versus the native forest approach, and I was wondering, in these calculations, do you take into account how long does it take for a new forest to be effective in terms of CO2 sequestration or biodiversity or the other parameters it had? What's the time scale?
Do you mean how long I calculate the timber or carbon sequestration?
I mean, for plantations, obviously they aren't really effective after 1 year, right? So how long does it take? And I guess you use eucalyptus maybe that grows very fast.
Yeah. So I selected. So we have different type of eucalyptus. So we have monoculture, and we also have eucalyptus mixed with other species. So for the monoculture, usually it's managed, the rotation is about 7 years. So we estimated the carbon sequestration or within 7 years. So for other forests, we estimated we take all the species restoration. Let me reframe that. We take all the species, the time we harvest, how long we harvest, take into consideration. So for mixed stand, probably the rotation is more than like 70 years. Yeah, I'm not sure I'm answered your question.
Yeah, wonderful. Thank you very much.
Thank you. So.
All right. We can discuss this more in the panel discussion if we have more questions. We will move on to, thank you, Dr. Jiang. We will move on to the next speaker, Dr. Youdong Mao. He's Boya-Endowed Professor, Center for Quantitative Biology from Peking University. Dr. Mao, please.
Okay. Okay. First of all, I'd like to thank the conference organizer to give me the opportunity to present our latest work. So my laboratory mostly focused on developing experimental and computational approach to investigate inner working mechanism of 26S proteasome from human. So the most interesting question we want to learn is how protein is degraded. We have some of results, although it's not off the story, that's presented in a current video. So we learned that the ubiquitin can be engaged with ubiquitin receptor and deubiquitinase, RPN11. And then this allows the substrate, which is represented by the red polypeptide, that can be inserted into a AAA ATPase. Then the AAA ATPase can undergo complicated conformational transition, which converts the chemical energy harvested from ATP hydrolysis into mechanical work, which unfold the substrate and translocate the substrate into proteolytic core particles. So what's interesting in this study is that we found that the ATPase motor, which is composed of 6 hexamer subunits that undergo 3 quite different distinct modes of ATP hydrolysis. The third mode, which is shown in the current slide, is one that we are most interested, which corresponds to processive substrate degradation. So in order to really understand how the substrate is unfolded and how the ATPase motor is controlled, we sorted to employ time-resolved cryo-electron microscopy and try to apply this approach to investigate the protein degradation process. So this will require 3 requisites. First of all, we need to prepare the sample in a time-resolved manner. This would really hinge on a successful in vitro reconstitution of reaction dynamics of USP14 controlled proteasome. The USP14 is another very important deubiquitinating enzyme that control the proteasome function. And because we were trying to imaging samples that in the course of chemical reaction before the substrate is degraded, so that we would expect there's lots of intermediate conformations, which will require development of more advanced 3-dimensional classification approach, which we were able to accomplish by introducing deep manifold learning. So through a combination of both experimental and computational innovated methodology, we were able to capture right in the middle over the course of the entire substrate degradation. And fortunately, we discovered several substrate engaged in intermediate states and also coexisting substrate inhibited in intermediate states. By tracking all 13 conformational states over the entire course of degradation, we're able to observe how each of these intermediates change over time. And this eventually give us the opportunity to observe a near complete cycle of ATP-driven substrate translocation when the core particle is fully activated, and as well as allow us to discover 3 kinetic checkpoints of USP14 regulation of proteasome function. The first checkpoint corresponds to initial ubiquitin recognition, where it can assign the substrate and ubiquitin to specific deubiquitinase. And the second checkpoint is when USP14 is activated, it can decide which substrate is degraded or rejected. And the third checkpoint is when the degradation and deubiquitinating activity is done, and when the ubiquitin chain is recycled, and all the 3 checkpoints would tightly regulates the proteasome function. These give us a deeper understanding of how protein degradation. Okay, thank you for your attention.
Well, thank you, Dr. Mao. This is open for comments and discussion. Yes, please, Dr. HERSHKO.
Yes. First of all, I would like to thank you very much for this very beautiful work of structure and function. I was of how the proteasome works, which is interested me very much. I was puzzled about one thing about USP14, the deubiquitinating enzyme. It can do 2 opposite things, as you pointed out. It has the substrate engaged pathway in which it causes the conformational change, which opens the gates to the ATPase, and the substrate goes in and is degraded. And it does also the opposite. The substrate inhibited pathway in which it releases, it cleaves the substrate, the protein from ubiquitin, from the ubiquitin chain, or at least for most of it, and then the substrate is not degraded. So I was wondering what determines the fate of the protein, whether it goes for degradation or it goes for release. Are there some differences maybe in the protein conformation, and what are these? Or maybe there are some differences in the ubiquitin chains. So what do you think about that?
Thanks a lot, Professor HERSHKO. That's a very interesting and important question that we believe we haven't been able to answer in a previous study, but we do have several hypotheses. First of all, we think the combination of the ubiquitinating enzyme and ubiquitin receptor form specific geometry that may impact how a substrate is engaged, or is it a happy lifetime to engage with the ATPase motor before USP14 close off the proteasome. So we believe that many of the substrate actually doesn't have enough time to actually engage with the ATPase to be sufficient degraded. So that effect we see is in general, USP14 is suppressing proteasome function. But if for certain substrate, it can really have a high rate of engagement with the ATPase motor, they can be degraded, and that's actually been captured in the substrate engaged pathway. But we also notice that once the substrate engaged pathway is completed for a single substrate, then it transits into substrate inhibited states. So it's like you have one chance of degrade one substrate, then at least before USP14 disengage, dissociate with proteasome, dissociate from proteasome, USP14 still impose stronger inhibition effects on the proteasome function. So that's kind of what we thought. So we like to, right now in our lab, we are generating different kinds of substrate that may have different geometries and different stability and different, so that they can have different chance of being engaged with the ATPase before USP14 even completely inhibit the substrate. So that's our current thinking.
Yeah. You know, when you are looking for from the biological sense point of view, you know, like we did many years ago, then you can see taking off the ubiquitin chain as a kind of correction mechanism. That is as if some proteins are ubiquitinated, but since they are not destined for degradation, they are being corrected. So the substrate inhibited pathway is a kind of a correction mechanism. On the other hand, you have the mechanism in which it actually helps USP14 helps protein degradation. So I guess by trimming it in a way that it should be suitable. So if you are looking from the biological sense point of view of the protein, then you would think that it must be something in the protein or in the way it's ubiquitinated that determines. So maybe it's worth looking at this point.
Right. Yeah. Thanks a lot for your suggestion. Yes. Indeed, we noticed that some of the substrate, their degradation were promoted by USP14. But in general, these proteins, we guess number of such USP14 promoted degraded substrate would be less than those that's being protected by USP14. But certainly biologically, these could be also important. Yeah, we will like to learn in the future. Yes.
So I would like to congratulate again on this beautiful work.
Thanks a lot. Thank you very much.
Do we have more comments or questions? Dr. Moser, please.
Yeah, again, not my field, but I found it interesting that you're using these deep learning methods for the classification of conformational states. So I was wondering, when you do that, what is actually the information you put into the model? Do you, for example, have to say in advance how many conformational states you expect to find, or is it completely open?
Thank you very much. And that's a very good question. So as I show in current slide, our approach, we're trying to use deep learning to reconstitute the pseudo energy landscape of the whole molecular system. So we do not need to predefine how many states are there. We just try to find how many local minimums are there. It's after we see this reconstituted energy landscape, then we start to kind of guess how many metastable states we need to reconstruct. So that's after we already process the data with machine learning. Thank you.
Wonderful. Thank you very much.
Can I ask you, very much related to the last question, how reliable are these results gained by the artificial intelligence?
Right. So in order to validate or verify the results, we have to, first of all, we have to collect sufficient, I would say, very large data sets to make each of the conformations reconstruction to be able to dump at 3 to 4 angstrom resolution range. Why we need such a high resolution for each of the conformation or conformer is because once we achieve near 3 angstrom resolution, then we can use all the stereochemistry information, which is not in any way used as input during the machine learning. So we use stereochemistry rules to validate the structures, right? For example, these reconstructions, you can clearly see the backbone distance and helical pitch and the beta sheets. All these informations were not given at the beginning of the classification. So after we classify them and reconstruct conformers at a high enough resolution, then we can use stereochemistry to validate them. As you can see that there are also areas where we have low resolution, we have higher energy. Those are the components we were not able to verify or validate. So we do not take structures from those areas for any of the biological interpretation at all. Thank you.
Dr. Andersson, do you have a question?
Yeah, I mean, looks like very beautiful work, and not in my area of expertise, but something which I'm very interested in is the relationship between genetic variation and phenotypic variation. So my question is, to which extent, I mean, this is a multi-protein complex, so to which extent does genetic changes in these cause disease in humans?
Right, yeah, very interesting question. So there has been other related works from which we also want to do expand our research in the future, is trying to Professor Wolfgang Baumeister from Max Planck Institute in Germany. They were trying to image proteasome in native neuron cells. So they try to see how these proteasome sample different conformations in situ. So they use cryo-electron tomography approach. So what they observed is there is a very good correspondence between the in vitro reconstituted conformations and compared to those conformations obtained from in situ cryo-electron tomography reconstructions of the human proteasome. So these allow us to be able to learn that if we actually can successfully reconstitute these structures in vitro. But of course, there's some kind of differences between resolutions achievable right now in for the cryo-ET methodology. But I think in the future, with all other related tools improvement, we might be able to see in situ proteasome reconstructions in neurons at very similar resolution as we right now can achieve for the in vitro reconstitutions. Thank you.
So my question was a little bit more that, so are there any inherited disorders that is due to mutations in the components of this? And if it is not, it could be because there's redundancy, that you could lose one of these RPNs and it's still sufficient function, or it's lethal, then you don't see any disorders either because they're never born.
Right. Yeah. That's true. So that's why right now, and I think previously, many of the proteasome subunits has been investigated by lockout or lockdown experiments. For example, the intrinsic deubiquitinase RPN11, it's once it's locked out, basically the cell is kind of cannot endure it. So it's very essential. But USP14, it's a totally different story. You can lock down or lock it out while the cell or mice is still alive. So yeah, they indeed, many of these subunits of the proteasome, they have kind of very different roles biologically. Yeah.
Okay, thank you.
Thank you.
Great. Any more questions? Dr. HERSHKO, your hand is still on. Do you have more questions? If not, we will move on. Thank you, Dr. Mao.
Thank you.
Next one will be myself. Let's start share. So in our lab, we study somatic-sensory mechanism. One thing that is pretty interesting to us is that how we process the each information. Each information processing is one of the sub-modality of somatic-sensory information processing. And that is probably the least known sensory modality. We sometimes can be bitten by a mosquito, and you feel this itch. And there are many interesting questions in this field. For example, why when you feel itch, there is a very strong desire to scratch, and sometimes you cannot control this desire. And when you scratch an itch, there will be some pleasure sometimes. What's the mechanism? That's something we're quite interested in. One thing is whether there is each specific gene or neurons that is really involved in the processing of each information. So early in our early study, we used this microarray analysis, and we did identify a gene that is called gastrin-releasing peptide receptor. That is each specific gene that is expressed in the spinal cord. And we found that we knock out this gene or ablate the neurons that express this gene, we will abolish the each signal transmission. And further, we asked whether there is some local gate in the spinal cord that can gate the transmission of each signal in the spinal cord. Now we identify that there's a group of GABAergic neurons in the spinal cord that play an important role in gauging the each information processing locally. The next question is how this each information is transmitted to the brain. What's the pathway? So from the neuron that express GRPR, we found that these neurons are not lung projection neurons. They are mostly local neurons. So we found that these neurons will synapse onto another neuron that is a projection neuron. This neuron will project to the brain area called paraparietal nucleus, and this pathway is very critical for sending each information to the brain. And if we suppress the activity of this pathway, the induced cognitive behavior will be suppressed. And we know that for the somatic sensory information, there's not only a local modulation, but also modulation from the brain. For example, there's anxiety, and it can cause enhancement of the itching sensation. So our question is that whether there are some neurons in the brain that play a critical role in the descending modulation. And we found that there are a group of neurons in the brain area in the brainstem, in the midbrain, that is critical for this descending modulation. This group of neurons express a peptide called substance P, and this group of neurons will send their projection to the spinal cord indirectly, and while this DISNAP disinhibition mechanism can amplify the each signal processing at the cell level. So now we try to understand how we distinguish the itch from other somatic sensory, for example, from touch, from pain. How we know if there's itch information, we know that's something that is itchiness. So with the photo microscopy, we measure the calcium response in the cortical area. We found that this is encoded with the population coding scheme, and we also discovered that the neural activity show that ignition-like activity when the animal feel itch, and this is happening in the primary sensory cortex, which means that when the animal feel itch, there is a group of neurons. They will fire together and will have the signal strong enough to propagate to other brain areas, and in that case will evoke the behavior response. Now this is our are good, but we try to understand the whole circuit network about not only about itchiness, but also about other sensory modality, especially somatic sensory modality. With higher resolution, for example, at the single neuron resolution. So that will provide a basic frame for our further understanding of the somatic sensory signal processing. With that, we use fluorescent micro-optical stacking tomography to map the single neuron morphology, and we can classify the spinal projection neurons into 10 subtypes, and further we can map the neurons of the downstream of the spinal projection neurons. That's how we can construct this wiring diagram that mediate the somatic sensory information. And this mapping of the connectome is actually a bigger project of microscopic brain connectome project that we are involved in. In the pilot study, we already constructed a single neuron resolution projectome of mouse hippocampus, the mouse prefrontal cortex, as well as the hypothalamus. And with the hope that we can finish this mouse microscopic connectome within the next 5 years, and will provide this basis for our understanding of mammalian brain. And the long-term goal is to map the mesoscopic connectome of non-human primates. Okay, that's all I'm going to share today. Thank you very much.
Yeah, I just wanted to first start by congratulating you in a very nice series of experiments. So as you say, each is really an studied phenomenon, and I think you are on a good way towards a better understanding of it. So I had two questions. So one is the obvious one, which probably a wider audience may wonder about, which is why actually does it help to scratch? Why does scratching take away the itch feeling? And so why does the behavior of scratching actually make any difference? And the second one is more related to how itching is perceived in the brain. So I think when you identified this receptor, gastrin-releasing peptide receptor, and saw that it is necessary for the scratching to occur, and you pointed to this connection to the parabrachial nucleus, and then further up to the somatosensory cortex. So I was wondering, is the itching, you showed then by decoding approach that you can tell from the activity in somatosensory cortex whether itching actually is perceived. So I was wondering, this should probably also be available in the parabrachial nucleus, and is this is probably harder to record. And I was also wondering about the somatosensory activity. Is there just one single state, or are there different types of itching related to the different outputs to the various brain areas that you showed at the end? There was 2 or 3 questions, but I hope you can elaborate on some of them.
Well, thank you, Dr. Moser. That's wonderful questions. So why there's this chronic behavior? Why not other behavior? For example, in response to the pain stimuli, there will be the withdrawal the behavior. So which is quite different from the chronic behavior. And evolutionarily, I think it makes sense because these two are both protective mechanism, and the withdrawal, you can stay away from the harmful situation. And but in some cases, there is something already attached to the skin or to the body. This withdrawal response cannot do that. You have to have another behavior to take away these harmful substances that are already on the skin or even getting the and how the time will stop itchiness. There's already study show that, as I mentioned, there is the local inhibition, and there's this early early hypothesis that there's a gateway theory. So there's activation of sensory fibers by the scratching. For example, the mechanical or pain sensory fibers that will probably will activate those inhibitors by this feed-forward inhibition. It will likely suppress the itch pathway that therefore will stop the itch signal from go on. And then the stimuli went away, and there's no more input, then there is no itch signal. And about the itch perception, I think there are for both pain and itch, there are two components actually. In recording from the somatosensory cortex, we are only targeting or only addressing the one component, that is sensory component. You know where it is, how strong it is, and maybe what kind of skin it is. The other component for itch as well as pain is the emotion component. If there's the pain stimuli or itch stimuli, you feel very bad. There's a negative emotion, and only these two combined together, you feel that it is itch or pain itself. I guess you are quite right. We want to record from other brain areas such as preparatory nucleus. So then we will have a full picture about the this very specific sensory modality. And yeah.
Just a quick follow-up to that. So I was wondering, the somatosensory activity is probably not necessary for the scratching to occur. I guess if you had inactivated somatosensory, it would it be sufficient that the signal went to the parabrachial or to the brainstem somewhere, and they could just go right back to activated scratching? Or you think that a more conscious perception is necessary?
I think both are involved because we actually have done this experiment that you proposed. So it will partially suppress this scratching behavior. So that means at least part of the behavior is conscious behavior, and but probably not all. So I think both are fully involved. Yeah.
Okay. Wonderful. Thank you very much.
Thank you, Dr. MOSER. Yes. Dr. Andersson?
Yes, so beautiful work, and so a reflection I had is that sometimes gene names can be very misleading. So you wouldn't expect that gastrin releasing peptide receptor would be controlling. I mean, it sounds so. But I think this illustrates when we're given gene names, it depends on the first function that we identified with the gene, and then later on we find that many genes are pleiotropic and has an effect in many different cell types. So, but my question was, so how did you identify this as the candidate for having this important role in regard to itching, the GRPR?
Yes, I think you are quite right. It was the original finding was not in the central nervous system. The way we identified this, as I mentioned, we did the microarray analysis. So the assumption was that the genes are involved in sensory stimuli will be highly expressed in the sensory area.
Okay, so you started with a gene expression study and identified this as a differentially expressed gene. So for that reason, you identified.
Yeah, okay, I'm sorry about this. So my question is about your needs in imaging. I mean, you showed beautiful 3D renditions towards the end of the talk. So what are your needs regarding imaging? So what kind of methodologies do you need, and what are the basic requirements for those methodologies?
I think there are two things are critical. One thing is that we want to label the neurons very sparsely. As probably this is old idea. The very early study by Kaha and Gorgi, they have done this. They label the neurons very sparsely with Gorgi staining. So here, that's one thing that is required. Otherwise, we cannot distinguish one neuron from the other. The other thing we need to image the whole volume of the brain. In this case, as I mentioned, we use this method of fluorescence micro optical sectioning tomography, and this has pretty high resolution, so we can reconstruct that. In that way, we can reconstruct this reject this volume of image, so we can see that we can clearly see each axon that is we can manually reconstruct. And we are now develop the automated approach.
I mean, how long did it take to take these pictures? I mean, so what is the recording time? For example, that zoomed out area.
I don't know. I can tell you about this. The whole brain, the whole mouse brain, it takes about 5 days. But for a small area, it should be probably hour or minutes. All right. If no more question, we'll go on to our next speaker, Dr. Zhou. He's professor of Department of Biophysics, School of Medicine of Zhejiang University. Dr. Zhou, please.
My work for the past 5 years mainly focused on the diversity of the oxidative phosphorylation in structural and mechanistic terms. So the idea of oxidative phosphorylation is, of course, proposed by Peter Michel, and he thinks that there are actually two kinds of protein complexes in the membrane of the mitochondria. One is called the NADH oxidase, which is responsible for the electron transportation, and the other one is called the ATPase, which is responsible for the synthesis of ATP. And these two complexes, they're actually energetically coupled by a cross-membrane proton multiforce instead of some energy absorbing molecules proposed previously. And later we find out that actually instead of one complex, we have 4 complexes, and they constitute the electron transport chain, and they kind of transport the electron sequentially from the NADH, which comes from the TCA cycle, to the ubiquinol in the membrane, and then to the soluble cytochrome C, and then to oxygen. So then you form water. And the structures of all these complexes are very difficult to solve in the crystallography era, but now we're in the cryo-EM era, so there's a boost in the structural studies of these complexes. What's more exciting is that instead of working individually, they also can form a supercomplex with a defined stoichiometry, like this resperazome, which is formed by 1 complex 1 and 1 complex 3, which is a constitutive dimer, and 1 complex 4. And there are also cryo-EM structural studies of these supercomplexes. And even more exciting is that we can couple these structural studies with the evolution of the eukaryotes, like the endosymbiotic event marks the beginning of the eukaryotes, and then we have the LICA, and then we have so many evolutionary branches of the eukaryotes. So the current structural study of the ETC mainly focus on the osbiocounts, which consists of fungi and the metazoan. And recently we've got a handful of structures from the Archaeoplastidia, which has the algae and the higher plants, Arabidopsis. But we are more interested in the last visited branches, such as the Abiolata, which contains the ciliates, and as well as the toxoplasma parasites. So then we use an important model organism, which is the Tetrahymena thermosomophila from the ciliates. Has been a classic model organism for the study of the ribozyme in the molecular biology. But then we use cryo-EM, investigated its electron transport chains, and found out that its electron transport chain is highly divergent compared to the classic mammalian or the fungi model. First of all, is cristae of the inner membrane of the mitochondria is actually tubular instead of the ridge-like cristae found in the osteocounts. So all the ETC complexes needs to adapt to this curved membrane. So we can see that there's actually a curvature for the membrane domain for each of these complexes. And secondly, is complex 1 and complex 3 form obligatory supercomplexes, which means that there's no free complex 1. And this is also different from the osteocounts, which they have the chance to be free or form into supercomplexes. And thirdly, is complex 4 is also highly divergent. So if we compare the classic complex 4 model, is actually a 200 kilodalton small subunit, a small complex. And now we've got a obligatory dimer, which is 2.7 megadalton. So which means that is more than 10-fold mass increase. And actually, we found 24 previously unknown subunits in complex 1. And for complex 4, we've got the 38, which includes 3 mitochondrial carriers. And we also have 6 homologs of a team chaperone domain. The team stands for the translocates on the inner membrane of the mitochondria. So these actually suggest that complex 4 in the Tetrahymena has actually evolved outside of the bioenergetics into a, I would say, bioenergetic hub and a scaffold for all the proteins on the membrane and the inner membrane of the mitochondria of different processes to attach to and utilize the proton motive force generated by this complex 4. Actually, a recent unpublished work from the group actually also reveals that these complexes can further form into even larger supercomplexes. We have or we can call it mega complexes. Actually, we've got 1 complex 4 dimer in the middle and 1 copy of the complex 1,3 obligatory supercomplex on each side of the dimeric complex 4. And then that large mega complex actually have a curvature of near 180 degrees. So it's a half ring-like structure, which is quite interesting to see. So apart from the electron transport chain, we also work on the ATP synthesis. So maybe I'll skip the basic mechanism and the structure of the ATP synthesis, but just to emphasize on what we found. So I helped to solve the structure of this prokaryotic V-type ATP synthase, and it differs from the mitochondria F-type ATP synthase by basically the number of the preferred stock. So you can see that for the mitochondria F-type, you've got 1 preferred stock. Prokaryotic V-type, we've got 2. Eukaryotic V-type, you've got 3. And also they differ, distinguished from each other by the composition of their central shaft, which rotates again relative to the rest of the enzyme. So the rest of the enzyme is stationary, but the central shaft will rotate. You can see that the mitochondria F-type actually has a 2-segment central shaft. But for V-type, we've got 3 segments. We've got this pink, yellow, pale yellow subunit D in the middle. So actually, that enables what we call the flexible coupling of the torque transmission of the V-type, because with this subunit D, actually the hydrophilic part of this enzyme can dissociate reversibly from the membrane part. And when they are not dissociated, when they are associated with the hydrophobic part, actually the unique structure design of the subunit D can actually prevent slippering between these 2 parts, which will rotate together, and then they will enable the torque transmission. This is kind of like the screwdriver and the screw. You can detach these 2, but when they are attached, actually you can transmit the torque. I guess that's what I'm going to talk today, and all this work are done in 3 institutes, Institute of Science and Technology, Austria, UC, University of California, Davis in the States, and the Zhejiang University here in China. And I will pay tribute to Professor Leonid Sazonov, who is the Amber Fellow, Royal Society Fellow, and Dr. James Ness, with a colleague and also a supervisor. Thank you for listening.
Thank you, Dr. Zhou. This is open for comments and discussion. Dr. MOSER, please.
Yeah, this is a naive question, but I still wondered. I mean, what are the methods that you're using to identify as discrete, so many discrete subtypes of the proteins and the complexes and supercomplexes? So are they so different that you can sort of tell by eye, or do you actually also rely on deep learning or artificial intelligence methods?
I guess currently there's not much study into the diversity of the electron transport chain complexes. So basically, we're studying one model organism of each of these evolutionary branches. So in the hope that since they depart from the LICA so early from other branches, so then their electron transport chain composition will be divergent, will be highly divergent compared to the others. So that has been proven successful till then. And another method that we are also employing is that we dig into the literature and see if there is morphological studies of the mitochondria. So usually if the membrane morphology of the cristae, the inner membrane, inner mitochondrial membranes are different, then the structures of these EPC complexes need to adapt to the different morphology. So this is also proven to be correct for the tetrahymena, because it's cristae is kind of tubular. So, but this is not always successful, because you can imagine, like for the osteocounts, the cristae is like the ridge of the mountain, but then at the actual ridge of the mountain, you've got very sharp bending of the membrane, but then at the two slopes, actually still planar membrane. So which so there's always a how do I say, sharp compartmental localization of these complexes on the cristae, which means that on the ridge, like for the osteocounts, on the ridge of the cristae, you've got ATP synthase dimers. So then the structure of the ATP synthase dimer can be highly divergent, but all the other electron transport chain complexes, they reside on the two slopes, which are not so different from others. And we are also trying to utilize genetic approaches to kind of predict the most divergent, the species that has the most divergent ETC compared to the classic model, but this is still very preliminary. So hope I answer your question.
Thank you very much. It's good.
I have another naive question. Do you have enough data to try to form an evolutionary tree? How these oxidative complexes evolved, from the early times?
Yeah, that's what we try to achieve, like to have a structural evolutionary tree, at least among the eukaryotes. But then
how close you are to that?
We are far from completing of that structural tree, and there are other groups working on the tree as well. And the idea we get is that all these are highly conserved. As long as it's aerobic cellular organisms, the structure plans for these are highly conserved, especially for the ATP synthase. So you can see that no matter how different their peripheral stalks or central shaft, they still have the same, almost the same plan of architecture, that they've got this serine in the membrane, they've got the alpha 3, beta 3 as the hydrophilic part. And the only subunits that differ would be what we call the accessory or the supernumerary subunits. But rarely you also see differences not conserved among the core subunits, because the bacteria complex, taking complex 1 as an example, so the bacteria complex 1 has only 14 subunits, 7 in the hydrophilic and 7 in the membrane. So those 14 subunits are called the core subunits, which is universally present in any of these species. But sometimes you've got slight differences, like for the tetrahymena, I've got 17 core subunits, which is more than 14. That's because some of the core subunits in the membrane actually split into 2 subunits. And So that's what we call the split core. But still, structurally, they topologically they overlap with the bacteria. But the number of the accessory subunits is actually very, very different among the different species. Like for bacteria, you have none. For mammalian, you have like 20-ish. For this Tetrahymena, you have additional 22. And the complex 4 is even more divergent compared to the mammalian version, because the mammalian version only has 14 subunits, of which only 3 are the core subunits. Here we of course have the 3 core subunits, but we have 38 and no accessory subunits.
Now the core subunits,
yes.
If they are derived from bacteria or to the mitochondria, then you would think that they would be all mitochondrially produced.
Yes.
Yes. But are they?
Yes, they are encoded by the mitochondrial genome.
All of them.
Yes.
Okay, so that fits. That fits.
Yes. Yes. And this also relates to the disease that caused by mutations in the core subunits, and most of them are mostly mapped to the mitochondrial genome, because if you have a mutation in the core subunits, usually you don't have the chance to survive. But then some of the disease like the Leif syndrome or the Mila syndrome, and you can survive until adolescence, I guess, but they are all mapped to the mitochondrial genome, which encodes the core subunits.
Yes, Dr. Andersson.
Yeah. So as you probably know, there are these initiatives these days to produce high quality genomes for basic. I mean, long-term goal is to have a high quality genome for all eukaryotes on the globe, and there are thousands of species that have high quality genomes. So would that be a resource for you to, because you could use, I mean, gather all the genes sequences from these and look at evolutionary pathways in different evolution lineages?
Yes, that's absolutely a resource for us. Actually, from time to time, we feel that resource is not enough for us, because one of our current projects actually also represents one of these evolutionary branches. But the challenge that we face is that there's no high quality genome for that species. But on the other hand, we are not geneticists, so we don't know how to obtain a high quality genome. Oh, yes,
yeah, so you know what you can. There are these initiatives to generate high quality genomes. So you could contact these groups that is involved in that and ask them to prioritize a species which is of value for research, so to speak. Because if they plan to do large numbers, they could prioritize one that is valuable for other researchers. So
yes, definitely, that's definitely something we're going to try. And one thing is that usually what we need is the high quality proteome that we can download from the UniProt, and genome is not enough for us. At least we need to have a high quality transcriptome. But our current experience is that we got the electron density, and we can somehow predict the sequence of each subunit from the high quality density. But then when you blast that query sequence against the available transcriptome that you have online, you can download. Usually you don't get any hits, and that's
okay. Yeah. Yeah.
All right. Okay, Dr. Mao, please.
Very nice work, Dr. Zhou. So I'm just wondering, because you're studying these complex composed of many subunits, and some of those hasn't been previously characterized. Have you guys been trying to use AlphaFold 2 to predict some of the structure before you actually start to build autonomy models? What did you find, if you have done so?
The AlphaFold has been proven useful to us in the sense that we use the AlphaFold structure as the initial model before we manually edit them according to the density. If you can identify the sequence of a certain subunit, of course you can predict the structure via AlphaFold, or if the sequence actually exists in the UniProt, you can directly download the AlphaFold structure from the UniProt, and then it's the question of rigid body fitting into the map and manually editing. according to the density, but that's all we use for the AlphaFold right now. And if you have a sequence that has a topological homologue in other species, that say the osteocounts that already has the structure solved, you don't even need to use the AlphaFold. You can just use the homology modeling of the Swiss model.
Okay, thank you. So just follow one more question.
Yes.
How similar did you find that your true structure model as compared to AlphaFold prediction?
Well, usually for the transmembrane helices, they predict with very high accuracy. But then usually we also have a hydrophilic domain. And for that domain, usually you can also predict with very high accuracy. But the relative position between different domains, like the TMH and the hydrophilic domain, usually the angle is not correct. So once you have the AlphaFold structure, you need to cut it into different domains and fit individually and then link them together.
No more. If no more question, that will be bring us to our the second part of our forum panel discussion. This panel discussion, our young scientists can have questions to answer for distinguished guests, and we can further discuss the presentation earlier or some other topics we might discuss.
I must admit, I didn't understand you. I had difficulties hearing you, actually. I don't know about the others. I didn't get your question or your point.
Your microphone is going a little bit on and off, in fact, so we lose you sometime.
Oh, okay, I'm sorry. So now we are going to start with the panel discussion. In this panel discussion, our young scientists can have questions to maybe can discuss with our guests, and we can, if we want, we can further discuss the early presentation or some other related topics. I see Dr. Jiang raised your hand. Please go ahead.
Okay, thank you, everyone. I have a naive question, probably, because I'm still at the early stage of academic career. So I'm specifically interested in what makes you a top scientist, and what do you deal with the challenges that arise from new methodology or technology? For instance, in order to estimate the global food production, you have to use a fresh new method to analysis. However, you are not experienced in this method. So how do you conquer this kind of challenges? It's an open question for everyone.
Okay, anyone want to? Yes, Dr. Moser, please.
Yeah, I can certainly relate to that question, and I can just say a few words about how this is in my own field in neuroscience, and especially in the neuroscience of behavior, because the techniques and approaches that existed when I was a student, and even when I was a young professor, are completely different than what they are now. That applies both to the way we acquire data. So, for example, up until quite recently, people essentially measure the activity from one cell at the same time and try to understand how the brain area works. Now, that is not the way to go any longer. I mean, we have approaches that can collect data from many thousands of cells at the same time and study that dynamics. With that then comes also a completely different ways to analyze the data, new statistical approaches, machine learning, artificial intelligence, as we heard in some of the other talks, and following that and being on top of it, doing it in your own lab is a hard challenge. How do I approach it? Well, I just try as best I can to collaborate with people who do it, and ideally then recruit people to my own lab who are on top of these fields. They are usually 20, 30 years younger, and for them, this is how they learn neuroscience. And then we work on it together as a team, and I try to understand it as best as I can, but because, of course, I don't have the training that a young neuroscientist today gets with coding and statistics and quite a lot of quantitative background that is needed. So I just support you, and this is a major challenge, but it also is very important to address in order to succeed, as you mentioned.
Thank you so much for the wonderful answer.
Okay, Dr. Hao.
Yes, I think you raised very important points. Your first question was, what makes you an distinguished scientist? Well, I think that applies to all of us. You have to be very passionate about what you do. And that's number one criterion. Regarding the methods, it very much resonates with what actually Edvard MOSER said a minute ago. If you look at the history of science, very often you will notice that the big paradigm change, and usually science evolves in paradigm change, not linearly, but there are the breakthroughs. Those breakthroughs very often are connected with methodological advancements or methodological breakthroughs. And that's why it's so important to stay up to date with methodologies, because usually it's the methodologies that define new changes in the coming years. There are exceptions, of course. I don't want to say there is no other ways of making breakthroughs, but very often, if you look at history, then you will see that totally new ways of measuring a piece of data made the fundamental change. That you have to keep abreast of new technologies.
Thank you. Passion, up to date, keep yourself updated. Thank you for
have the courage. I've had the courage to adopt the new technologies, because I fully understand there is a hurdle, as Edvard said. So it's of course very nice to stay within your own say bubble or work with methodologies that you're acquainted with, but you'll be in a year or two you'll be completely left behind, because if a new methodology can do something that is much better, you'll be left behind. I give you one example. Now, say 7 years back, say the resolution in super resolution microscopy was 20 nanometer standard. Now the standard is 2 nanometers. And those who will not adapt, yeah, I'm sure there are a couple of them that will not adapt, will be left behind, because those that adapt, they can get information the others don't.
Okay, thank you, Dr. Hao. Dr. HERSHKO.
Yes, I have a little different view. I think some discoveries are driven by new methodologies. Sometimes they're not. Always driven by a good question. So first of all, you should ask a good question that is not known. For example, I guess as you asked about how optimally to manage your forest. Then you have to make sure that you are not in the mainstream. Otherwise, there is no. You should do something new, not the mainstream. I think that's important. And then you should find how to approach this question. Sometimes it's by a new methodology you can learn. Sometimes it's by using known methodologies. But I think the most important thing is that you will ask a new and important question, and that you will make sure that you are not in the mainstream, that you are doing something new, that you are trying to form a new stream. So that is my advice to you.
Thank you all for this very valuable input. Thank you so much. I've learned a lot. Thank you.
Okay. Dr. Mao, do you have some questions?
Yeah. I have a question specific for Dr. HERSHKO.
Yes.
Because our field is focused on protein degradation, so I have a similar question that what do you expect that what's your kind of imagination of the future of the landscape of protein degradation? What's the biggest issues you would expect to be solved. Thank you.
Well, as you were, can you hear me? I'm not sure if
Yes, yes.
Yes, okay. So of course you did wonderful work on the proteasome, but before the proteasome there is the ubiquitin ligases. And I think the main questions in protein degradation are selectivity and regulation. So what selects a certain protein for degradation and certain rate, and what regulates that? So I think there are some ubiquitin ligases about which a lot are known. For example, like the APCC that I am working on, there's some beautiful work of structural biologists and biochemists and cell biologists. But there are some others who do very important work. So I would think that ubiquitin ligases is a big field, and there is still a lot to do about it. And of course, there are the myriad functions of ubiquitination. It's not only protein degradation, but many other things, signal transduction, DNA repair, and so on and so forth. So I think ubiquitin ligases and their roles in different pathways of ubiquitination. That's still for the future.
Okay, thank you very much.
Dr. Zhou?
Yes. I have a general question to ask all of our honorable guests. It's like, how do you cope with the challenge when you transit from a postdoc to an independent scientist? This maybe happens decades ago for all of you, but this is the current situation for me, and I'm finding this transition stage a little bit challenging, because suddenly publication is a little bit more difficult without the supervisor's name on it, and the grant application is something that is quite totally different from the publication of scientific papers. So do you have any advice for the new PIs like me and maybe Dr. Jiang?
Dr.
Yeah, if I may say something, I found the transition from postdoc to PI quite difficult and quite challenging. And it took me a while until I managed to adapt for many reasons. Also because, I mean, if you have a small group, usually as a postdoc, I work with just 2-3 people, and then I'm at a group of 2-3 people or so. Then you're like a friend, a very close friend to everyone, but then all of a sudden you have to take responsibility, and then you have to be a boss as well, and supervise somebody and give grades and marks and the rest of it. And then you phase out of this transition of being like a pal, a friend or whatever, and become a boss. So that's a big psychological transition. For me, it was very difficult, I must admit. So it took me years till I managed to deal with that. The second thing is, sure, I mean, grant applications and the rest of it, financing labs, takes more than writing good grants. Very often, and that is my experience, it also takes a lot of networking within the community that kind of is your peers, and they kind of judge your work. And that is sometimes very subjective. And because it's subjective, it takes very good relationships with these people. Now, I'm not doing this anymore. I'm in the Max Planck system, and I don't write grants. The last grant I wrote is maybe 20 years back. So, and that's why I'm so happy that I don't have to do that. And because I don't have to do that, I can speak very openly about it. I don't even review grants. So if I ask somebody to review a grant, I'd say no, thanks. I think grant reviewing is a very tricky thing, and I have not the best opinions about it, to be very honest with you, because it's very subjective. And I can go on at length at that. I'm not saying one should not write grants, so don't get me wrong. I don't want to make black and white situations. There's positive things about writing grants because it forces you to think, forces you to kind of clear out in your head what you want to do. But there are also the dark sides of grant writing, which is the social and all the implications of this. So I think one has to have both stable funding that is with no strings attached and perhaps also some sort of grant writing. But it's tricky. And I agree with you, it's tricky.
Yes, I think that's it's a very important question, and there isn't any quick fix really. But I think one of the things that I try to say to people who I have advised is that it is important to not lose track of the big questions, because that goes back to the first question we had, namely what makes you a successful scientist. And I think that is very much about the questions you ask. Also, to follow up on what Dr. HERSHKO said, I mean, that defines how successful you are, whether you ask a good question that matters for many people and which drives science forward. But those questions usually don't have an answer within 3 years. So then I see the challenges of publication pressure and all that. And I think to some extent, this is really also up to the institution around you, that they have to give you support to allow you to at least spend a major part of your time on things that take a while to solve. I also wanted to say one other thing that I quite often observe, which relates back to grant writing, because sometimes some people just get too many grants. And that's also a problem, because it has a risk that your group may grow too fast. And I think it is important to grow slowly and steadily, and especially in the beginning, because you need to know what's going on in the lab. You need to be there and have, at least in the beginning, when you don't have reliable people around you, to have a hands-on feeling of the data acquisition. And I think then it is grow slowly, but not expand into a mega group all at once. That is usually not good. So that would be my two pieces of advice.
So if I may add something in simple terms, and Avram HERSHKO said that first here in this round. Of course, you can see the creativity of a scientist. You can instantly see it from the question that he addresses. Just look what he does or she does. That also tells a lot, not everything, but a lot about the creativity of that person.
All right. Dr. HERSHKO, your hand is on. You want to have some comments?
Well, postdoctoral period is a very important stage, you know, in the development of a scientist. He is kind of semi-independent, and that's an important part, you know, so that he is getting out of the tutorship and beginning to use his own or her own wings. So that's a good part. And it's still, they don't have to worry about grants. They will worry about it later. The problem today is that the postdoctoral periods are too long to my taste. You know, they used to be 2-3 years, and now they're 5, 6, 7 years. And by the time, because they have to publish in these high-profile journals, by the time they are finished, they are getting old. You know, some people. Somebody once looked at all the Nobel Prize winners, and most of them their discoveries before the age of 40. You know, and if you spend so much time in your postdoc, then by the time he begins to be independent, he's more than that. So I think we all have a responsibility not to use the good postdocs for too long. I think we have to release them as soon as they are. You know, they are beginning to be independent. I don't know if that answered your question, but that's my thoughts about the postdoctoral fellowship period.
If I may add, of course, you can also make a discovery as a postdoc, yeah. But of course, then you have to rely on the lab head to be fair to you and share the accolades with you, or you have to share it with him or her. So that adds to the complication. Of course, it's always better to make discoveries as an independent person. So I was not totally independent when I had my great ideas. But the person in charge of the lab, he recognized that it was my idea, not his. and he wasn't on the paper. Yeah. So I'm saying it depends very much on the culture. It also depends on the lab head and the rest of it. But generally, I fully agree with Avram HERSHKO. It's if you're creative enough and you're destined to make a career in science, the better you get the sooner you get independent, of course, the better it is. If you have the resources for becoming independent. And long postdoc times are definitely not something that one should do.
Okay, Dr. Andersson, can you unmute yourself?
Yeah, I see. Okay. Yeah, so I agree very much with what others have been saying here. And I mean, I think the simple answer is that young investigator need to find his own way or her own way. I mean, a risk is, of course, if you work too much in the footsteps of your supervisor, it could be negative for your development. So, but something I think what we have developed in Sweden, a quite good system, is to try to identify the most promising young scientists and give them a good starting ground so they could develop their own ideas and to get 5-year funding, not just 3-year funding, so you have time to develop your ideas. I think that is a very important aspect. And I'm curious to hear if there is a similar system in China that is supporting young investigator to develop their independence.
Yeah, we do have our starting grants, which can support, if you spend the budget like fairly and carefully, can support like 3, 4, 5, even 5 years. But then you're also under pressure to acquire funding as soon as you join the institute. That is something that everybody needs to do here in China.
I think it's important to have a little bit longer starting ground, like 5 years, because it takes time to establish yourself. You don't have to just go for a secure project. You can try some really interesting ideas.
Okay, I have a question to ask our distinguished guest. I think you are all very successful in your career. And what do you consider to be the biggest challenge during your scientific career, and how did you overcome this?
If I may answer this question, so when I started to work as a scientist, I identified at that time very unique question: can I overcome the diffraction barrier in a light microscope? And I was the only one who tried to do it in a light focusing microscope, in a microscope that used just lenses. And this was considered so crazy and outlandish that of course I always had to ask myself whether I'm on the right track, yes or no. And if you're just the only one in the world, yeah, and of course there is no recognition from outside. You have difficulties publishing in visible journals. So my first papers were in very unknown journals. There was no resonance, and then of course it takes a lot of, how should I put it, self-confidence, courage, intuition, whatever, to stay on track. That was difficult for me, and I can tell you in the very early days, sometimes, of course, I thought whether I'm totally on the wrong track and the others are right. Fortunately, I was lucky in the end that the Max Planck Institute that I work now became aware of me and gave me a 5 years contract, as we've just heard, as a young scientist, to work on this was considered at that time crazy problem. And then after I had first indications, after 3-4 years, of course, they even hired me as a director, although I've never had any nature, science, or physical review letters paper. I didn't have any of these flashy papers, but they just looked at the potential and the originality of the work that I worked in, and then gave me the chance to continue and to develop it. So so the difficulties I had initially were actually to keep on moving despite the fact that there was no positive feedback from the community or negative feedback for a long time. Actually, the most influential people at that time, they even went around saying that my data is perhaps not reliable and these kind of things. They wrote negative reviews when I applied for jobs at universities. As I found out later, really bad things. And you have to have stamina to continue and to through that period. That can be very hard.
So when you ask yourself whether you are in the right way or wrong way, do you have some kind of a backup plan? What if you are wrong?
Actually, that's a good point. So when I started here as a young scientist, my 5-year contract, I had 2 projects: the high flying project, which won me the Nobel, and I had a safety project, because I didn't know whether that high flying project would work. And the safety project worked. And actually the community at that time said the safe project, which wasn't, in my view, scientifically not that interesting, was the great work. They told me, this is the great work, not your high flying stuff. This is not useful. Yeah, it won't work. It's not you. But the safety project was good. And when I was appointed director, I was not appointed because of the safety project, because of the potential of the others. The people recognized that, my colleagues around here. I instantly dropped the safety project, instantly. I said, I'm not going to do any safety project anymore. I have now a tenure job. I want to go just after the big project. And I dropped the rest and just went after this big project. So I think there's an advantage of having a certain security, tenure, or whatever, because then you can follow the really interesting projects, those that are risky, those that have high return if they work out. But of course, this situation can also be abused. I could have said, no, I have now a tenure job, I'm doing other things. It depends on the personality, if they take this chance or not.
Yes, so my answer would be related to several of the questions we have discussed already. So my challenge as a scientist was to fund my research. So when I started my research career, like in the 80s, so that was when molecular genetics was developed, that we could start to do deep genetic research, genome research, and so on. And my idea was that we could use the tools of modern genetics to understand how domestic animals have been domesticated. What type of genetic changes have happened in the domestic animals to transform a wild boar into a domestic pig, or a red jungle fowl into domestic chicken, as a way to increase understanding of the genotype-phenotype relationship. But I had this problem. I couldn't get this research funded in Sweden, because if I went to the Agriculture Research Council, they thought this was basic research. They were not interested to understand the history of evolution of domestic animals. If I went to the Natural Science Research Council, they said, well, this type of work shouldn't be done in pigs or chicken. It should be done in Drosophila or in mouse. So they rejected my grant as well. So if I had stayed, I could easily have been not able to fund my research at that time. So what saved me, and once I submitted my research to this research council, and I got this comment, and then I was so angry because I felt that the reviewers didn't understand what I was saying. So I called them and asked them, who is reviewing this research grant? And it turned out that these were very well-respected scientists. But I think what that illustrates is our inability to, even if you are a well-established scientist, you not always understand the ideas behind another scientist. I think very much what Stefan was talking about, so I think even if you are a Nobel Prize winner or whatever, you need to be humble, I think. So I think it's the most important, try to understand the creativity and whether it's important questions that the person is trying to understand, and not be and be realized that you not always understand what you should. But what saved me was that research on domestic animals had a higher priority in the European Union. So I got European funding and to establish my lab, and then I got Swedish funding later on. And I got the Wolf Prize on this work. So,
thank you, Dr. Andersson. Dr. HERSHKO, yeah, you go ahead.
Yes, Yeah. I guess you heard two people, and then if you will talk to many people who achieved new results, you will see that the common denominator is obstinacy, to be obstinate, to stick to your guns, not to leave your aim because people don't like it. So I think that's very important. So don't care about what other people say. Just do what you think is important. And I think I have to go in 5 minutes. So
okay. Yeah.
Okay.
Dr. Moser?
Yeah, what I wanted to say is somewhat related to what Dr. HERSHKO mentioned. So I think what accounted for at least some of my own success is that I always followed my own intuition, partly because I didn't really know very much. I mean, I started out from a background, non-academic background. I didn't really have many people to ask, but I just knew what I wanted to do. So the positive thing is that I could make my own choices and not depend on others. The negative thing is that by not having a huge network around me, that actually didn't show me the options. So I think in retrospect, I can say that I would probably be a lot faster if I had known the many various choices I could have made and directly go to the right labs and so on, instead of taking many detours in the beginning. But I think I got the right networks in the end, and I think this is very important to as a young scientist, that you team up with people who have good connections and can help you being exposed to the world at the start when you're not very famous yet, and then which gives you more opportunities. At the same time, as I must say again, and which then also relates back to what Dr. HERSHKO said, that I think one should only listen to advice to some extent, but not fully, because then you won't be successful. It is what you bring on your own confidence about where you want to go that may make the difference.
Thank you, Dr. Moser. We're almost at the end of this forum. Anyone who want to make more comments or questions to this summit? Dr. Jiang, please.
I would like to take this valuable chance to ask Prof. Andersson a specific question. So because you are a very knowledgeable in food production, so I would like to ask from the point of view of ensuring food production. So how can we manage our forest? The rationale behind this question is there's a trade-off between food production and timber production. Actually, the agriculture expansion is one of the main reasons caused forest loss. So although our government have did some things and have spared some land for instance, to produce food, and spared some land for produce timber, but this seems not enough. So from your point of view, what we can do more for ensuring food production and timber production?
Yeah, I mean, this is of course a grand challenge for the future for all people on Earth. That is this conflict in land use, how much we use for forest production, how much we use for agriculture, and I think an important message is that I think we need to be aware that we need to change lifestyle in general in the world, and one way is of course to reduce consumption of animal, so that we should avoid to take down forest to produce food for animals. So it's much more welfare-friendly way to eat more plant-based food. I think that is very important. In fact, I took part in a panel discussions on Saturday evening in the WLA, and I took up two. So that was in fact food security and agriculture. So I took up two aspects which I think is important. One is that I think we also need to discuss population growth in the world, because we could certainly, I mean, I'm 100% for that we should improve technology to improve the production of food and make it more efficient and so on. But I think we still need to be concerned about the rapid population growth on the Earth, which is because that drives this conflict in land use to how much is used for buildings, how much is used for agriculture, how much is used for forestry. So I think that is an important question we need to consider more. And something which I took up, I think there's a lot of areas where we are not as efficient as we. The thing I took up, something I work with these days, is in fact the biodiversity of fish. So what we are doing at the moment is that there's a lot of species that we, instead of eating the fish, we catch the fish and fish meal to feed other animals. So feed for aquaculture, salmon production, and we feed also fur animals and chicken and so on. And that is a waste. You may lose 90% of the animal protein and energy in that conversion. So we could utilize our marine resources in a more sustainable way. That would also be a way to increase food security in the world. And you know, when they fish this species, often clupids and others, pelagic fish, the price for that fish is 20 cents per kilogram. So this is a very cheap protein resource which could help to reduce starvation in the world. So I think there are many aspects we need to consider to reduce this conflict. So
thank you so much for the answer.
All right. Thank you for this question, and I think food safety is obviously very, very important. This is obvious, especially during this given this Ukraine war. And this comes to our end of this session. This session we cover a lot of topics, including biodiversity, structural biology, as well as neurobiology. We have discussed about especially the career development and somewhat on the paradigm shifts and education. I think this is all important to learn for, especially for our young scientists. I personally learned a lot. I think the other three young scientists will probably agree with me as well. At the end, on the behalf of the organizing committee, I would like to thank all the laureates for joining us to share your thoughts and experience with our young scientists. And I also want to thank our young scientists for joining this forum to share your latest new discoveries. Also want to thank the audience for coming to this forum. Thank you. That's all. We will close this for this session. I think we are just right on time. 1 minute before this. Thank you.
Thank you for very nice presentations. Bye-bye.
Bye.
Thank you for you all.